Hey everybody, I'm Chris, and this is episode 2 of That's Mother Science. Traditional models of intelligence have been primarily based upon rough estimates of brain size or volume. This is motivated by the notion that larger volume equated to more neurons and thus more computational power. In 1983, a paper entitled Manual for Raven's Progressive Matrices and Vocabulary Scales utilized PET, positron emission tomography. Yes, that means they shot antimatter into people's brains. Found an inverse correlation between metabolic activity and test scores, which were the metric for intelligence. This evidence was in stark contrast to the popularized model of brain volume. This has been further corroborated by studies utilizing data acquisition via PET, fMRI, and EEG, as stated in the paper reviewed today. The major study discussed here relies on NADI. For info on this approach, in the description is linked to an open access article. There are also some references to the Pfit model, which indicates potential brain regions as candidates for future studies focusing on morphological differences related to intelligence. This paper was published in 2007 and it is not open access. Regardless, a link will be provided in the description to this video. This paper, self-reported as the first study utilizing NADI to study microstructural architecture in the brain. This was published in Nature Communications, and for once, something published by Nature could be accessed by poor people. To all the students over the summer that give a shit, they used a clever approach. They compared NADI imaging to the 180 brain region divisions developed by the Human Connectome Project. Data was acquired on volume, neurite density, which was signified by INVF, neurite orientation dispersion, or ODI, and isotropic diffusion, ISO. These measurements were signified as both from the cortical tissue and white matter, the wiring. Two sample groups were used. The first group, S259, whose name reflects its size, used the BOMAT written test as the metric for intelligence. The second group, S498, was named likewise, utilized a separate test, the PMAT24. In sample S259, neurite density and neurite orientation in the cortex negatively correlated to scores on the BOMAT. In sample S498, similar findings. In partial correlation analysis, for sample S259, volume of the cortex related to the BOMAT score. Volume of the white matter did not show any correlation. In sample S498, interestingly, a negative correlation on neurite orientation in the white matter was shown with PMAT24 scores. Researchers ran an analysis of single brain regions in an attempt to corroborate the Pfit model. In the first group, sample S259, they found that 159 out of 180 regions for neurite density and 164 out of 180 for neurite dispersion regions showed negative correlation with intelligence scores. Partial correlation analysis showed that neurite density possessed negative correlation for 12 regions. Interestingly, 9 out of 12 regions overlapped with specific brain regions designated to be candidates in the Pfit model. In the second group, S498, neurite density in the cortex showed negative correlation in 154 out of 180 regions, and neurite orientation showed 151 out of 180 regions possessing negative correlation. In partial correlation analysis for the second group, for neurite density, 11 regions showed negative correlation, 7 of which overlapped with the Pfit model. And for neurite dispersion, 5 regions correlated negatively, 4 of which also overlapped with the Pfit model. This data as suggested by the author, that efficiency in computation would be an indicator of intelligence. A schematic was presented showing relative neurite densities and connectivity in low IQ and high IQ individuals. These findings offer compelling evidence that intelligence is facilitated by higher levels of synaptic pruning, leading to more sparsely connected yet more efficient networks.
And that's mother science.